Um, Thanks, everyone. So I'll just introduce myself. My name is Tommaso Di Ianni. I am an assistant professor in psychiatry, and I have an appointment in radiology. I direct a basic ultrasound research program here at UCSF, and I was awarded a PARC Fellowship this year. And thank you to the Pain and Addiction Research Center for the generous support and for supporting our research. So I'm going to talk about our work in ketamine and specifically mapping ketamine effects in the right brain. And we are particularly interested in network effects and how these are mediated by opioid receptors and by biological sex. Many of you may know that ketamine, that there is a lot of excitement in psychiatry for ketamine, psychiatry and neurology. And in particular in psychiatry, the reason is that ketamine has rapid antidepressant and anti-suicidal effects, which led to the FDA approval of S-ketamine, one of the two stereoisomers, for treatment-resistant depression in 2019. However, we still don't know exactly how ketamine is working. The textbook definition of ketamine is that it is an NMDA receptor antagonist, but there is emerging evidence supporting potential involvement of the opioid receptors, specifically in the antidepressant effect of ketamine. And the implication of opioid receptors may have consequences in terms of abuse liability and potential for dependence. So the question is that us and many others are trying to address is whether opioid receptors do actually mediate ketamine's antidepressant effects. And some studies have shown that when we block the opioid receptors, the antidepressant effects of ketamine are suppressed. This is in particular something that two studies from 2018 and 2019 from Nolan Williams at Stanford have shown when they were pre-treated the animals with naltrexone, an opioid receptor antagonist, before administering ketamine. The antidepressant effect of ketamine was nearly abolished nearly completely. However, there are other studies, including one from UCSF, that have shown no significant effects using opioid agonists and antagonists. So this is a question of open debate. So our approach has been to reverse translate some of these experimental paradigms. In particular, we are referring to the experimental design of the papers by Nolan Williams that I've shown before, but we're reverse translating these paradigms in rats so that we can map map what ketamine is doing in the brain in rats, but we are using modalities that are translatable in nature. In particular, we are excited about functional ultrasound imaging, and this is a relatively new neuroimaging modality that provides outcomes, provides a readout that is very highly correlated to the bold signal in fMRI. So the idea here is that we are taking some observations from the human side, we are reverse translating them into animals. We are doing functional ultrasound imaging in addition to other manipulations that I will show that are more invasive, but then the readouts that we get, hopefully we can feed them back into the clinic. And that's what I mean with a two-way translational pathways, because we have this kind of feedback. So in the setup that we used for this study, we had only access to 2D imaging. So we're imaging brain slices. However, we are moving away from this, and I will briefly talk about this later in the presentation. So we had to decide where to look, and we focused on two brain slices, one anterior one and one more posterior, like posterior to bregma. And the reason why we picked these slices is that there are many brain regions that are very interesting for both depression, for substance use disorders, and that have been also previously implicated with the mechanism of action of ketamine. So that's where we decided to look at. Now I will show a video of what happens in real time when we administer ketamine. So we are imaging the brain continuously, and there is a timer here. And so we just administered ketamine at the 0 time. And you can see that there is a very rapid rush in surge in neural activity in the prefrontal cortex, in the retrosplenial cortex, another region that has been also implicated with ketamine, and specifically also with the dissociative effects of ketamine, and also some activation in the basal ganglia. And these effects peak at around 10 minutes, so these are transient. And they are almost completely resolved between 40 and 50 minutes post-ketamine administration, as we would expect, since this overlaps with the pharmacokinetics of the drug. So when we blocked the opioid receptors, we found some very interesting results. So on the top here, so here I'm showing some statistical parametric maps. So here we are comparing rats in the naltrexone plus ketamine condition to rats in the vehicle plus ketamine condition. And these were actually the same rats that we imaged repeatedly, and the order was randomized to minimize carryover effects. And so the two interesting observations from these images. The first one is that there are definitely many regions where blocking the opioid receptors seem to either attenuate, and that's shown as blue-green color, or enhance, shown in red and yellow, the effects of ketamine. And these regions include all these interesting regions that I mentioned earlier: the prefrontal cortex, in particular the cingulate area 1 of the prefrontal cortex, the nucleus accumbens and caudate putamen, so the dorsal and ventral striatum, the retrospinal cortex, and the lateral habenula. There is another region that is very interesting, and probably I'm assuming Elyssa is going to talk more about in her presentation. The other interesting observation is that these effects were only limited to male rats. So on the top row here, we are seeing male rats, and on the bottom, we see the females. And you can see that in females, we have nearly no significant clusters. And these results are confirmed by looking at the time series that I'm showing here. So again, this is the cingulate area 1 of the prefrontal cortex, the caudate putamen, and the nucleus accumbens. And you can see that if you compare the blue trace, which is vehicle plus ketamine, to the black trace that is naltrexone plus ketamine, you can see that in all these regions, naltrexone is partially suppressing the response to ketamine, but that is only the effect for males. And males are on the left side, females are on the right side. So in females, we have virtually no effects, at least at first sight. So the next question is whether this is an effect that is specific to ketamine, so especially the opioid-mediated responses, if it is specific to ketamine, or is it just by way of doing an injection? Like if you do the injection with and without opioid receptors on board, there might be like confounding effects associated with that. And so we repeated these experiments with MK801, which is an NMDA receptor antagonist that is much more selective than ketamine, and it binds to the same site at the NMDA receptor as ketamine does. And in this case, we did not see any differences between the two conditions. So as you can see here, both in the bar plots, if you compare the first two bars are vehicle plus ketamine versus naltrexone plus ketamine, and the other bars are vehicle plus MK801 and naltrexone plus MK801. You can see that there are essentially no differences between these two conditions, as you can also see in the time series. The next question that we try to address is whether these effects are mediated, and the sex, and I'm talking about the sex-dependent effects, are mediated by neurodevelopmental differences between the male and female brain or by sex hormones. So we removed the gonads in males where we did see an effect in the first place. We waited for testosterone to wash out. We repeated these experiments. You can see that the effect is completely gone. So this speaks to like a gating effect of testosterone that is then kind of enabling this somehow, and we don't know exactly how yet. It is enabling this kind of a compounding effect between naltrexone and ketamine. We did not limit ourselves to looking at imaging. All those measures are very acute. So in these plots that I'm showing here, we are quantifying a protein that is called PSD95. It's postsynaptic density 95, and it is a marker of structural plasticity. So it is something that we know already from the literature that after we administer ketamine in rats, we have an overexpression of this protein in the medial prefrontal cortex. And so this is what we see here, both in male rats on the top and in female rats. You can see that there is a very significant increase in the vehicle plus ketamine condition compared to the vehicle only condition. So ketamine is increasing this protein as we expect. However, when we treat the animals with naltrexone before administering ketamine, this effect is completely gone in male rats. However, in females, it is not completely suppressed. There is still some residual expression, but it is non-significant. It's not significantly different compared to the vehicle plus ketamine condition. We also looked at locomotor sensitization. So in this case, we are administering ketamine repeatedly over 4 days. And as you can see, both in males and in female rats, there is an increase in locomotor behavior. So this speaks to potential adaptations in the dopaminergic system. And we are completely blocking this locomotor sensitization when we pretreat the animals with naltrexone. As you can see here now, if you compare the blue plot to the black plot, but it appears only to be the case, again, in males and not in female rats. So wherever we looked, both in terms of acute measures and more chronic measures, and also with repeated administrations, everywhere we found these differences, both in terms of when opioid receptors are available versus when opioid receptors are blocked, and also in males versus females. So more recently, we became interested in looking at functional connectivity changes. So in all the data that I've shown before with functional ultrasound imaging, we were only looking at differences in activation between these different regions. So now we are looking at how these regions talk to each other. So we're looking at circuit and network level effects that we are, again, quantifying with functional ultrasound imaging. And so here we are segmenting these brain regions, and then we correlate each brain region to each other, and we obtain these correlation matrices as the one that I'm showing here on the right side, where we have this full picture of the brain, and in this case, it's just a single slice of how these different regions are correlated with each other. And when we administer ketamine, now if you compare the matrix in the center, that is 10 minutes post-ketamine administration, if you compare that to the baseline on the left side, you can see that there are some definite effects, especially in the insula. So on this bluing effect here, that is all in the anterior insula and granular and dysgranular insula. So pretty much with everything else, everything becomes decorrelated in that region. But also you can see that there is a decorrelation in these functionally homologous contralateral brain regions, as you can see on the diagonal here. And also there is a decorrelation of within the medial prefrontal cortex. So the prelimbic and infralimbic cortices, they appear to be at baseline decorrelated from everything else, but highly correlated with each other. And ketamine has some effects in kind of decorrelating these brain regions bilaterally. So all of these, they were all kind of very interesting effects for us to find. And in these plots here, I'm showing some of those regions where we have statistically significant effects. And as again, you can see the medial prefrontal cortex here and the insula on the bottom. And one thing that I would like to stress here is that in most of these ROI pairs, we see a decorrelating effect of ketamine. However, there are also some cases, like in this case within the medial prefrontal cortex, where we do see an increase in correlation following ketamine that is then again normalized at 40 minutes post-ketamine administration. Now what happens, again, going back to opioid receptors and what we see when we pretreat the animals with naltrexone. So I would just ask you to compare the matrix on the right side, and that is the naltrexone plus ketamine condition at the same time point as the vehicle plus ketamine condition in the center. And so if you compare the right side to the baseline, you can see that is a lot more similar than in the middle. So this indicates that at least at the first degree of approximation, naltrexone is somewhat counteracting the effect of ketamine, but importantly, naltrexone alone doesn't give us any statistically significant effects or interactions. So that means that naltrexone, at least in this brain slice, appears to be essentially inert. It's not like we're just averaging out the effect of ketamine. This speaks to a more complex interaction between ketamine and opioid receptors and sex. And if now we see these plots here on the right side, these are kind of breaking down some of these effects in these regions where we see statistically significant effects. Again, this is in the prefrontal cortex. We also see some effects in the somatosensory cortex. And this is still something that we're trying to understand, but it is very interesting that, so the orange plot is the vehicle plus ketamine condition. And you can see that in all these regions, at least ketamine is decreasing these correlations, okay? In females, we have some effects, but they're not quite as strong, and in these cases, they're not significant. However, when we pretreat with naltrexone, the effect in males is almost completely abolished. But in females, we see an opposite effect. And this is very interesting because in the previous work that I've shown before, one of the things that we found using autoradiography is that there appears to be a compensatory effect in females when we block the opioid receptors. And again, we don't know exactly how this is working out, but it is interesting that with a completely different modality, at different time points and everything, we are seeing effects that are going in the direction of this potential compensatory effect that is happening in females when we block the opioid receptors. Again, importantly, on the right side, you can see here that naltrexone alone in and of itself is almost inert, is not really changing these functional connectivity measures in the rat brain. Another thing that we're looking at, we became very interested in network level effects in terms of networks that have been identified in the brain using functional MRI in humans. And one in particular is the default mode network that is heavily implicated with major depressive disorder. And also there are reports that ketamine is modulating the connectivity within the default mode network, within nodes of the default mode network, and also between the default mode network and other regions. So we wanted to kind of see what happens in this case. Unfortunately, we don't have the whole brain, but we do have one important node of the default mode network that is the medial prefrontal cortex. So in this case, we are segmenting the medial prefrontal cortex. We get one temporal signal that we then correlate to all the other pixels in our slice. So now we're getting kind of this spatial distribution of the correlations between this seed and everything else in these slices. And you can see that the baseline is very similar between males and females. So on the top row, again, we have males. On the bottom row, we have females. When we pretreat the animals with when we administer ketamine, we have this decorrelating effect between this node and everything else in the brain. But when we pretreat with naltrexone, that effect is nearly completely abolished in males. However, in females, we do have this decorrelating effect, but it stays the same between the vehicle plus ketamine and naltrexone plus ketamine conditions at the same time points. And these here are, again, statistical parametric maps comparing at the group level these different conditions. And again, you can see that there are these strong effects within the medial prefrontal cortex. So it is decreasing partially the correlation within the seed, but it is increasing significantly the correlation with other nodes of the prefrontal cortex, in particular the cingulate area 1. And this is in males. In females, we don't have much going on. And then when we pretreat with naltrexone, so this is what we're seeing now here at the bottom, we see also these effects within the node, so inside the prelimbic cortex in this case, but also with another region that is the dorsal striatum, so they call it putamen. So these are all kind of effects that are interesting to us, and we're trying to understand what these mean. But it is interesting that we have this kind of a global decorrelation that naltrexone is somewhat preventing. And we see here also, again, here in these statistical parametric maps. Now, these are network level effects. But another thing that we're looking at is trying to investigate what happens at the local level. So we're using, for this, we're using the Kendall's coefficient of concordance. And this is a measure of local synchrony. So we're not in the data that I've shown before, we're correlating this seed to everything else. In this case, we don't have any seed, which is a nice thing because we don't have to pick a seed. So it's more data driven. And then we are calculating this Kendall's coefficient of concordance in a very small kernel, and then we run the kernel over the images, and so we calculate this coefficient for the entire images. And you can see that we have a decrease in KCC with upon ketamine administration. It is somewhat blocked by naltrexone, and it's also evident in females. And it is very evident when we look again at the prelimbic and infralimbic cortex, so the medial prefrontal cortex node that we looked at previously, and also in the caudate putamen. And this is very interesting because we know from other reports that have other studies that have reported these effects with ketamine alone, but also with other molecules like nitrous oxide, LSD, and so on. So what we're showing here is this sex difference and the fact that these effects are dependent on the availability of opioid receptors. So in conclusion, we're very excited about dig deeper into these effects that we found, both in terms of opioid-mediated and sex-different effects at the network level, and we're moving more progressively towards the local and circuit level interactions. And we're really excited about trying to understand how these two connect with each other. So how these local effects, kind of how important they are they causally implicated with the global effects, or is it just a correlation? So this is something that we are addressing right now. And thank you again to the Pain and Addiction Research Center for the support. So with that, I would like to thank my lab, our collaborators, including Dr. Josh Burke for the continual mentoring and support, PARC and the Pritzker Family Foundation for enabling us to do this research, and thank you all for your attention.
Great, thank you, Tommaso. So I think we probably have some questions, hopefully, from people. In the meantime, maybe you can illuminate me on a couple of things, just because of my lack of background knowledge here. So when we think about the signals that you're measuring, how should we think about these signals compared to other modalities like fMRI signals, and BOLD and so forth? And then within the context of depression and human depression studies, are there functional connectivity analyses that we should be directly relating to the functional connectivity observations that you're making?
Okay, yeah, so to answer your first question, people have actually looked at it. There is a study that I actually cited by Edelman, so it's from the lab of Jean Lee at Stanford, and they looked back-to-back at functional ultrasound imaging and fMRI while they were stimulating the motor cortex with optogenetics. So they have a very clean stimulus in both cases, and they compared both modalities back-to-back. And what they showed is that they are very highly correlated. So the readouts are very similar between the two modalities. As you would expect, they're both based on neurovascular coupling. But also functional ultrasound imaging appears to be a lot more sensitive. And I think the reason for that is that we're averaging less. So our resolution is higher. So we're in fMRI, we're potentially averaging more kind of counteracting effects. So we may be averaging out some of those effects, while with functional ultrasound imaging, because we have a higher resolution, we can retain some of those. So we have ultimately a larger power and larger effect size. In terms of functional connectivity in humans, there is a very broad literature about these measures. Now, one thing I want to say is that in all these cases, we're looking at very acute times, at least for the functional ultrasound imaging. So what I'm thinking is that these measures are actually more, and this is still kind of work in progress, but I'm thinking that these are more kind of indicative of the dissociative effects of ketamine, or like the kind of the experience. But I think that is very interesting to us because there is, for example, work by Boris Heifetz at Stanford that they have given subanesthetic ketamine in a surgical setting. I mean, of course, there are many confounds there because the patients are anesthetized. So they're not only receiving ketamine, but they're receiving a lot more. But in that case, they had a huge placebo effect, and they didn't find any differences between ketamine and the control molecule. So kind of, this is to speak to the fact that there are some kind of, essentially, I don't want to say that it's all placebo, we don't know, right? But I think these measures are more kind of indicative of those specific effects, kind of more related to the dissociative effect. And also with the Kendall's coefficient of concordance, that's the same. Kind of there are studies that have been investigating those measures and kind of in the context of altered states of consciousness, for example. So with LSD, with psilocybin, with ketamine during sleep, for example. So in all these different situations. So that's, I think, kind of what we are looking at here. It's more kind of relating to these kind of these time points, essentially. It depends on time when we look at it.
Okay. Time for some questions from the audience. Just unmute yourself if you can.
Yeah, I don't have any idea. Actually, I wanted to follow up on Josh's question. Now, ultrasound's been around for a long time in terms of imaging. Functional ultrasound, I just have no idea how that works. I mean, are you looking for a sound that's emitted from the blood vessels, or is it looking for some changes, in density? What is it that you're doing there? Are you beaming a sound in and looking for a rebound like in sonar?
No, we are, well, yeah, I mean, in a way it is like sonar, meaning that it's all pulse echo, and that's all ultrasound imaging. We
Pulse
send
echo,
ultrasound
okay.
in and we
All right.
listen what comes out.
Okay.
So essentially what we're doing here is blood flow imaging. So we are measuring blood flow,
Yeah.
which is something that has been around forever. But the reason why only recently we can do functional ultrasound imaging is just that because of a technological leap that came from the fact that now we are using we're offloading a lot of the processing into software. So basically we have GPUs to do that, while before we didn't have GPUs. It was a lot harder to see these signals from these tiny blood vessels. It's a more of a technical issue that was essentially solved about 15 years ago. And so now we are able to process all these data because we have large data sets. So we're able to process all this data and look at these tiny blood vessels. Otherwise, in the brain, we would only be able to, let's say in the human brain, we would only be able to look at the circle of Willis. So only like very major blood vessels, but we don't get any functional information from those blood vessels because they're averaging out again like a lot of signals, a lot of blood signals from different brain regions that might be in opposing directions. Now, in terms of what we are seeing here, more specifically, the signal that we're measuring is cerebral blood volume. So in both fMRI, we look at the ratio of oxygenation and deoxygenated blood. In this case, we are essentially looking at the number of red blood cells that are crossing the resolution voxel at any given time. And in and of itself, if we take a screenshot, it doesn't give us any functional information. But just like fMRI, when we track these signals in time, we can infer the functional information, because what's the underlying principle is neurovascular coupling, which tells us that the brain has no intrinsic capacity to store oxygen and glucose. And so if there is an increase in spiking activity, there must be an increase in, and I mean, I use there must be with, quote unquote, because of course this is also something that is heavily studied, but there appears to be an increase in blood flows. And so if we track those blood flows, we can indirectly infer neural activity. And that is exactly what we're doing here.
Okay, thank you.
Welcome. Thank you for your question.
Time for one more question, perhaps.
Hello, I have one. So I've never worked with a ketamine IV. So with rats, what's the behavioral state of the animal? So if you were to try to pick it up, could you flip it onto its back, or if you hold it by the tail, does it shake?
You mean when we administer ketamine?
Yes, yeah.
No, I mean they're kind of wobbly a little bit.
Okay.
I mean, this is in the open field I'm talking about, when we do it in the open field.
Yeah.
They do wobble a little bit, but they're not, this is like we're using 10 milligrams per kilo, which is a dose that has been widely used in the literature for kind of the antidepressant-like effect of ketamine. And so we see the animals wobble a little bit, but they're not fully anesthetized, they're not fully down. And within, I would say, 15 to 20 minutes, they go kind of, you wouldn't see the effect anymore, but we do see locomotor, like increases in locomotor behavior when we administer it. So at first they go down, but then when they come out, they do run around more, like significantly more than before or than other.
Okay, so it's like first you get the suppression, but then they come back and they're more active.
Yeah, I mean, it's not, honestly, we never quantified it. So this is more like, me looking at the animals when we do the experiment. So just with, with a grain of salt, I would take it with a grain of salt. But yes, I mean, there is a very acute phase that lasts a few minutes. when they are still awake and maybe walking around, but kind of a little wobbly, and then they come out of it and they run around more
So
than.
it could be,
yes.
it could be interesting to try and do exactly that correlation and see which aspects of connectivity and activity correlate with those locomotor
Absolutely.
on a moment by moment basis.
I was
That'll be good.
just about
Yeah.
to suggest that.
Absolutely.
Great idea.
Yeah,
that
we
sounds
are
good.
looking at these
Yeah.
measures, yeah, and also doing it more chronically, like looking at longer periods of time, which we can do because these systems can be, the animals can wear it. So we can potentially look at kind of extended periods of time and see
Okay,
how
my
these.
apologies, Tommaso.
No,
my
sorry,
apologies.
yeah, I talked
we
too
need
much,
to,
sorry.
we need, no, not enough, but we have very limited time, I'm afraid. So we need to move on to Elyssa. So thank you again.
Thank
for
you,
your
and thanks
very
everyone.
interesting talk. All right, so now I'm sorry I wasn't here before, or got dropped before, but now it's my pleasure to introduce Elyssa Margolis, who's a full professor in the Alcohol and Addiction Research Group within the Department of Neurology. Go ahead, Elyssa.
Great. Thanks, Josh, and thanks everybody for coming today. And I'm going to tell you a little bit about our ongoing work studying how opioids control the lateral habenula, especially in the context of pain and pain relief. We first got interested in studying the lateral habenula because there's a decent amount of information in the literature indicating that lateral habenula neurons, the majority of them, fire more in response to all different kinds of aversive stimuli and aversive experiences. They also have higher firing rates in models of depression. There was also good evidence in the literature that stimulating excitatory glutamatergic inputs to the lateral habenula was very aversive. The lateral habenula is a pretty small brain region. This is a coronal section of a rodent brain, and it's sitting here between the dorsal hippocampus and the thalamus. And it's broken up into two major regions, the medial habenula that's characterized by this very dense population of neurons, and the lateral habenula that has many very tiny subnuclei that's a little bit more diffuse. And there was also some provocative information in the literature that maybe opioids also had an important role in modulating pain relief in the habenula. And in this particular study from back in the 90s, they showed that when they were just looking at acute responses to noxious heat stimulus, microinjecting a mu agonist or morphine really into the periaqueductal gray that would delay the animal's response to the heat stimulus, and putting naloxone into the habenula was sufficient to reverse that effect of the morphine modulation of the periaqueductal gray, suggesting not only was the lateral habenula or the habenula involved in responding to this acute noxious stimulation, but something about opioid receptors was contributing to the antinociceptive effect of the periaqueductal gray modulation. So we designed a series of studies, some are published, some are unpublished, where we have been looking at how mu-opioid receptor activation in the lateral habenula contributes to pain relief and the neural circuits that are involved. And I'll give you a little bit of a taste of some of our findings here today. So the first big question for us was, does activation of the mu-opioid receptor in the lateral habenula relieve ongoing pain? For this, we selected an ongoing pain model of spared nerve injury, and we cannulated the rats with directed at the lateral habenula so that we could microinject the mu agonist dam go directly into the lateral habenula. And we measured both somatic responses, nociceptive responses, and we used a condition-place preference paradigm in order to evaluate the affective component of the ongoing pain. And in these measurements, we found that in the place conditioning paradigm, when we microinjected the mu agonist DAMGO into the lateral habenula in sham animals that didn't have ongoing pain, we did not see any behavioral impact. But in animals that did have ongoing pain with the spared nerve injury, we got a conditioned place preference consistent with relief of that ongoing pain. When we looked at responses to mechanical stimulation with von Frey fibers in the sham animals, activating mu receptors in the lateral habenula did not impact their responses. But in the animals with the SNI, we did see a reversal of the allodynia from the SNI injury. In females, we did see a bit of a different pattern. The dose of DAMGO that relieved the ongoing pain in the males was not sufficient to do that in females. We had to go to a higher dose. And then we did get conditioned place preference in the females that had pain, again, not in the females without pain, but we didn't see any impact on the sensory responses in the females, even with this higher dose. So we know that from these experiments that activating mu opioid receptor in the lateral habenula is contributing to the relief of the ongoing pain. How could this be happening at the synaptic level? The simplest hypothesis for us was that since activating lateral habenula neurons appears to be aversive, if mu opioid receptor activation inhibits the neurons or decreases activity in the lateral habenula, that should drive pain relief. And so mechanistically, that would be either inhibiting the cell bodies of the lateral habenula neurons or inhibiting the excitatory glutamatergic inputs onto the lateral habenula neurons. And so with slice electrophysiology, we did indeed find that DAMGO, the mu agonist, inhibits the excitatory inputs onto a subset of lateral habenula neurons. Importantly, in the context of pain, that those responses were still there. We also saw in a subset of neurons, this is now a voltage clamp experiment, so an outward current or an inhibitory response is in the upward direction. In a subset of neurons, we did see a DAMGO-induced kind of modest outward current in response to DAMGO. So both of those are potential activity sites for mu opioid receptors to decrease the activity in lateral habenula neurons. So which inputs are these? In order to identify this, we put the optogenetic channel, the light-sensitive channel, channelrhodopsin, into several different brain regions that we know project to the lateral habenula and release glutamate there. And that way we could select out specifically the input we were looking at, we were interested in, and test if any of them were inhibited by DAMGO. We looked at a variety of inputs, including a couple that are very strongly associated with signaling pain and pain relief. But to our surprise, the strongest mu inhibition, which would be down by 0 here, were the glutamatergic inputs from the lateral preoptic area of the hypothalamus, rather than from the ventral pallidum, entopeduncular nucleus, lateral hypothalamus, or ventral tegmental area. So a little bit of a surprise for which input seemed to be mu responsive. With this information, we went back into the animal to test if manipulating this particular circuit from the lateral preoptic area to the lateral habenula could generate pain relief in animals with pain. So the way we did this experiment is we had animals with spared nerve injury, and we put an inhibitory opsin specifically into the neurons that project from the lateral preoptic area to the lateral habenula. So with light, we could turn them off, and in a conditioned place preference assay, turning off the neural connection in sham animals did not have any response. But in animals that had the ongoing pain, the SNI, inhibiting that connection did produce a condition-place preference, very parallel to the effect of the DAMGO when we micro-injected that into the lateral habenula. So one interesting thing that we saw is that neurons that received input from the lateral preoptic area also tended to have a postsynaptic mu-opioid receptor response. So if there was a glutamatergic input from the lateral preoptic area onto that lateral habenula neuron, we would see that neuron tended to also have an inhibition directly from the mu. Compared to the lateral habenula neurons that received inputs from these other regions. And if we looked at the strength of those inhibitions, there was a nice correlation between the inhibition of glutamate release. So again, here, these individual squares are neurons. Neurons that had a stronger inhibition would be close to 0 on the x-axis, and a larger hyperpolarization would be have a greater measurement on the y-axis. And so neurons that had a strong presynaptic inhibition also had a stronger postsynaptic inhibition, suggesting that these mu-opioid receptors are very well targeted to both pre- and postsynaptically control a very specific circuit through the lateral habenula. And so our next question was, where do these mu-sensitive neurons project from the lateral habenula? The lateral habenula has many potentially interesting projection targets, especially in the limbic system, but one really caught my eye for these pain questions, which is the parabrachial nucleus, because I wanted the simplest circuit in my mind was something that was going straight to a brain region that contributed to canonical pain responses. And one important question is, since it's such a small brain region and a relatively small population of neurons, do those neurons that project to these different outputs for the lateral habenula, do they collateralize to multiple brain regions? There's good evidence in the literature that the neurons that project to the dorsal raphe and the VTA are separate populations of neurons, but what about these neurons that project to the parabrachial? To answer this question, we started by looking at, we put a retrograde tracer in the parabrachial nucleus and in the dorsal raphe nucleus. And when we look back in the lateral habenula, the majority of neurons that are labeled by the retrograde tracer in the parabrachial are not collateralizing to the dorsal raphe. So this is only one of the projections, one of the non-parabrachial projections of the lateral habenula, but this could be a very specific population of neurons. When we record from the lateral habenula neurons that specifically project to parabrachial, we do indeed see a very nice hyperpolarization in response to mu receptor activation. And here I'm comparing our parabrachial projecting neurons to lateral habenula neurons that project to the lateral dorsal tegmentum. And we see a very different distribution of mu-opioid receptor responses there. And so for this work, one of our next steps is to test whether these, we have this circuit from lateral preoptic area to lateral habenula to parabrachial, if that is a connected circuit, and also to take this information of this on this projection from lateral habenula to parabrachial, go back into the animal and behaviorally test if this contributes to the signaling of ongoing pain that's coming out of the lateral habenula. And the final experiment that I want to tell you about today is something that we're really in the middle of analyzing, which is what is the distribution of mu-opioid receptors across the habenula. So as I said earlier, the habenula is quite small, but the medial habenula and lateral habenula are anatomically distinct, not just in how they appear with cell densities, but also with the circuits that they participate in. And there was a very provocative study from Brigitte Kiefer's lab several years ago, well, 10 years ago now, where they made a mouse where every mu receptor is fused to an M-cherry fluorescent reporter. So when you see the fluorescence in the brain slice, those neurons that show the M-cherry should have the mu receptor. And this is an example of their slice through the habenula, showing kind of basically a hot spot right through this border between medial and lateral habenula. And I think it's really important to see exactly where those neurons are. Like I said, the medial and lateral habenula participate in at least partially separate neural circuits. And so figuring out which ones might be most controlled by mu and how those contribute to addiction behaviors and pain relief behaviors, I think, are pretty critical. So just to tease you with a little bit of data, here we're collaborating with Corey Harwell's lab here in the neurology department. Rihanna Simon in his lab is doing this work for us. We're doing RNA-scope to detect the mRNA for OPRM1, the mu-opioid receptor gene. And here these slices are through the anterior to posterior extent of the habenula. And the DAPI marker shows you the density for lateral habenula versus medial habenula. And at the gene expression level, we definitely see a nice density of neurons expressing OPRM1 that are on the medial side of the medial lateral habenula border, but also quite a few neurons that are spread out in the lateral habenula that also express OPRM1. We're also looking at protein using immunocytochemistry against the mu-opioid receptor. And this is work that Ashka Popat in my lab is developing. And so this way we can not only see which neurons express the receptor, but exactly where they're localized, including if they're localized in fibers or even axon terminals coming into the lateral habenula, which is our expectation given what we've seen with the inputs from the lateral preoptic area that I mentioned earlier. And here, again, we do see an intensity of mu-opioid receptor protein right at the border between medial and lateral habenula, consistent with the mRNA, but also quite a bit of fibers that are seen on the lateral, especially fibers seen on the lateral habenula side of the border. And here's a little zoomed in image to give you a better idea. So we do see a relatively small population of neurons that seem to express mu on the medial side of the medial lateral divide, as well as quite a bit of fibers in the lateral habenula that are expressing mu. And so we're working on mapping these out, both the protein and the message. So together, I've shown you some of our findings related to mu-opioid receptor modulation of the lateral habenula and how it's contributing to pain relief. We are also, again, looking at exactly where the mu-opioid receptors are expressed and how they modulate these different neural populations and different inputs and outputs of the lateral habenula. And our working hypothesis right now is that lateral preoptic area inputs to a specific subpopulation of lateral habenula neurons is particularly strongly inhibited by mu receptor activation, and those neurons project to the parabrachial nucleus. And we hypothesize that this is a critical way that a noxious pain signal gets out of the lateral habenula. So to close, I want to thank my lab and our collaborators who've been working on this project, especially Maggie Wong, who worked on the published part of this work that I described today, as well as Corey and Rihanna for their collaboration here, and Ashka for her work that I talked about today. And I'd also like to thank the San Francisco Painless Research Foundation, who have helped to support some of this work through PARC. And I'm happy to answer questions.
Thank you, Elyssa. So are there other properties of this pathway, LPO, habenula pathway, that present opportunities for potential therapeutic interventions in terms of their receptor expression and so forth?
Yeah, we don't have an answer to that question specifically for the LPO to lateral habenula pathway yet, but there are several orphan GPCRs that are expressed in subpopulations of lateral habenula neurons, and very specifically in lateral habenula versus medial habenula, or even other brain regions. So developing ligands that target those orphan GPCRs may be a great way to access this specific circuit without the side effects that we run into with opioids.
Great. Other questions from the audience?
Just one. Is there any evidence for where the endogenous mu-acting opioid peptides might be? Are there any, let's say, enkephalin-expressing neurons in the lateral habenula or in the lateral preoptic?
There's a tiny bit of evidence that there might be some proenkephalin neurons in the lateral habenula, but it seems like a very small population of neurons. There could be proenkephalin inputs from the ventral pallidum and maybe some parts of the hypothalamus, including the LPO. One very provocative input in my mind is the input from the lateral hypothalamus, which does project to a very similar part of the lateral habenula as these LPO inputs, but those LH neurons express orexin and dynorphin. And although we think of dynorphin as a kappa opioid receptor agonist, it actually has very high affinity for the mu opioid receptor. So it's plausible that even that could be participating in this effect.
Thank you. Good answer.
Other questions? Let's go ahead and unmute yourself if you have a question. In the meantime, so whenever I see these sort of chains of circuits from one to the next, I'm always wondering what computation is being performed rather than just relaying information. What does the literature tell us about that or possible computations taking place?
Yeah, so one of the things that I'm very interested in thinking about how the lateral habenula works is that pretty much every neuron in the lateral habenula is a glutamatergic neuron. And there are quite a few local synapses, glutamate neuron to other glutamate neuron. And we have a little bit of preliminary data from our whole cell slice experiments that suggests that these mu-expressing lateral habenula neurons are one of the major sources of those local glutamatergic connections. So in addition to sending a sort of lined connection through the lateral habenula, I do think that there's a possibility that these LHB neurons could be distributing that signal to other projections as well, even if the LPO is specifically synapsing onto LHB neurons that project onto the parabrachial nucleus.
Okay. All right. Any more questions from the audience?
How about the delta receptor?
We have not looked at the delta receptor yet.
Okay.
Yes.
Okay. All right. Just giving a few more seconds for people to pipe up with questions. If not, I want to thank Elyssa again, thank both our speakers, Tommaso and Elyssa, for very interesting, if too short, talks. So, thank you both again.